So, hi, I'm Dr. Sarah Ivory, and I am an assistant professor at Penn State University in the Department of Geosciences. And what I do is that I am a paleoecologist. And so, if we break that down, what that means is that I'm an ecologist, and so I study interactions between different parts of the environment. That's what an ecologist does. But when you add the paleo to it, paleo means old, and so what that means is that I study ancient ecosystems, so ancient interactions within the environment in the past. And essentially, why I'm trying to do that is to try to better understand what are actually the causes of changes in ecosystems so that we might be able to better predict how ecosystems might change in the future in the ways that we're altering them today through climate change as well as through things like the growth of cities and through farming. And so I use the fossil record in order to try to reconstruct those ancient ecosystems, and in particular the plants that used to exist in the past in different places. And the tool that my group uses in order to do those reconstructions, so to know about ancient ecosystems, is actually microscopic. It's the fossilized pollen grains from flowers that get preserved in places like at the bottom of a lake in the sediments. And so we go and we collect those sediments, and then we're able to extract the pollen from the sediments and identify the plants at different levels in a sediment core in order to know what plants lived in a place at a certain time. And so that's essentially what a paleoecologist does, and what my group does for their research. And then we also are mostly interested in the tropics within my group. And so we work in places that, a lot of people never get a chance to even see. And so we have this sort of lucky focus where we get to go to a lot of really interesting places like Eastern Africa and Southern Arabia, like places like Oman. And that's where most of where we work.
Hi everyone, my name is Katie. I'm a PhD student here at Penn State University, and I work with Dr. Sarah Ivory. You heard Dr. Ivory just now explain a little bit about what our lab group does and what paleoecology is. So now I'm going to take you through a little bit about what I do for my PhD research here at Penn State. So I work at the southern end of the Arabian Peninsula in a country called Oman. This is located near the equator, and it's very hot, very dry climate. But despite it being very hot and very dry, it's a biodiversity hotspot. What this means is that there are a lot of plants that live in the region today, and there are a lot of plants there that don't occur anywhere else in the world.
Okay, so I explained to you a little bit about where I work in the Dhofar region of Oman, and that it's a biodiversity hotspot. And as you all know, global warming is affecting our planet. And we want to know what happens when you warm the already warmest places on Earth. And to do that, we can look to the paleoecological record. And as Dr. Ivory explained, we use pollen. But pollen is usually deposited in, say, lake sediments or marine sediments. But what do we do in a region like Oman, where it's really dry and there aren't any lakes? And for that, we can use hyrax middens. So you may be wondering, what is a hyrax? So rock hyraxes live in Dhofar, and they inhabit rocky cliff shelters. And this is the rock hyrax here. I call him Frankie. And you may be thinking, well, this looks like a rodent, maybe a groundhog. But they're actually not related to rodents at all. In fact, their closest living relative is an elephant. And so they inhabit cliffs, and they use communal bathrooms that are separate from their dens. And they accumulate as layered deposits, and they have a crystallized urinary product called hyrax, which is their pee. So as they're foraging about and collecting pollen and eating plants, the pollen can be brought into these caves on their feet or fur, or pass through their digestive system and end up stuck in the crystallized urine and the fecal pellets. And it accumulates as a midden, which is basically just the toilet. So here's a close-up of a midden, or a rock hyrax bathtub. So you can see some of the poop pellets here, and they're caught in this hard substance, which is the hyraceum. And so the pollen gets trapped in the hyraceum as the hyraxes pee, and it crystallizes or solidifies, and it can preserve pollen and other paleoenvironmental proxies for thousands of years. So what do we do with the middens after we've collected them from these caves and shipped them back to Penn State? The first thing we do is we want to rehydrate it. So we basically take the midden and put it in tubs of deionized water. And this allows the hyraceum to rehydrate until it's no longer a solid midden. And so we take this liquid solution and we want to treat it with a bunch of chemicals because there's a lot of stuff in there. We just want to look at the pollen. So we could treat it with acids like hydrochloric acid, which dissolves carbonates, or hydrofluoric acid, which dissolves silicates. And so through these chemical treatments, we isolate the pollen into its own solution. So we're in Dr. Ivory's new lab that's currently being set up here at the Dyke Building at Penn State. And I explained to you a little bit about what we do to treat the raw hyrax mids in order to isolate the pollen for further data analysis. And so it's important that we do this work in this contraption, which is called a fume hood. And what this does is it makes sure it circulates the air so that when we're using dangerous chemicals like hydrochloric acid, we aren't breathing in the toxic fumes. We also use things like a hot bath, which will gently warm our samples if we need to heat them. And we can also use this big thing, which is a centrifuge. And so we can put a solution inside the centrifuge and it'll spin it at a very high velocity. And what this does is it separates out components of the solution that are different densities. So when we're done with all of the chemical analyses to isolate the pollen, we're left with a little vial that has pollen and a little bit of alcohol left over. But what we want is to add glycerin, which is a common ingredient in lotions and moisturizers, and we want to get out that alcohol that's left over. So we put it in this contraption, which is called a desiccator. So the vials with the pollen and the glycerin and the alcohol are left in these trays, and it will quickly evaporate off the alcohol, from which we can remove that and take the pollen and the glycerin and mount them onto microscope slides for pollen analysis at a microscope.
Okay, so we were in the lab and we concentrated our pollen and extracted it from the raw hyrax mids, and we had it in our solution of glycerin, and we've taken it now to the microscope room. And so here we take the solution out of the vial and we can put it, a little drip of it, onto a glass microscope slide and cover it and look at it under the microscope in order to start looking at pollen. And the glycerin is really important. It's a thick substance, and so it sits between the glass slide and the cover slip, or the cover of the microscope slide, and the pollen inside is able to float and be suspended. So instead of being stuck flat on the slide, it's floating a little bit, and we can move it around in order to look at different parts of the pollen grain, which is three-dimensional.
Here are some nifty diagrams that show us how we can identify pollen grains based on a couple of different characteristics. First are the number of apertures, or openings from which the pollen germinates. And so these apertures can be pores, which are little holes, or furrows, which are shown here in the diagram as the lines. And depending upon the number and combination of pores and furrows, that tells us something about the pollen grain. So for example, triporate would mean 3 pores, whereas tricolporate would mean that it had 3 pores as well as 3 furrows. So another defining characteristic of pollen grains are their textures. And so these pictures here show what different pollen grain textures look like from the side, as well as when you're looking straight down at them from the microscope. So for example, in the third row in the first column is echinate. So these are like spikes. Okay, so we have our sample and I'm looking at it under the microscope and I found a pollen grain. And so I can look down the oculars here and use this knob, which if I turn this or this way, I can zoom into or out of the screen or out of the slide and look at both the apertures and the texture in order to identify this pollen grain to a specific type of plant. And so based on the texture and the apertures, I know that this pollen grain comes from a Vernonia plant. And so Vernonia lies within the Asteraceae family of plants, which you may commonly know as the daisy family. And so it looks flat here on the screen, but this is actually a 3-dimensional pollen grain. This is a 3D model of that pollen grain that shows you that spiky texture and the different apertures, the different pores. Palynologists, or scientists who study pollen and can identify pollen, can use a nifty tool called a reference collection. So all of these glass slides here contain samples of pollen that have been preserved and put in these cases, so that when I am having trouble identifying pollen grain, I can come here and pull out a slide and know the family and the genus and species of that pollen type and see if it matches the characteristics of the pollen grain that I'm looking at.
Hi everyone, so I'm back in my office in the Dyke Building. I just showed you what a pollen grain looks like under the microscope, but in a real hyrax midden sample, I would come across many different kinds of pollen that I would have to identify and then count how many that I see. So my data set looks like different types of plants and then how many pollen grains occurred in each sample. And so I have many different midden samples that are different ages, and so that allows me to reconstruct what the vegetation looked like on the landscape, so what plants were there and their abundances relative to one another, and how that changes through time. So in addition to pollen, there are other paleoenvironmental indicators or proxies within the middens, different kinds of chemicals that can tell me a little bit about what the temperature or what rainfall conditions were like at the time that the midden was deposited as well. And so I can combine that information to see how the climate is changing, so how temperature and rainfall are changing, and how the vegetation changes based on the pollen data, to get a sense of how changing climate may have impacted the landscape and how vegetation changed in response to changing climate. And if you remember, that's one of our main goals. We want to understand how climate changed in the past and how vegetation changed in the past to better understand what the impact of modern global warming will be on this biodiverse vegetation. So I've had a really fun time making this video for you all. I hope you enjoyed it, and thanks for listening.
Hi there, my name's Caitlin, and I'll be telling you about how you can be a scientist at home, just like Katie. So Katie told us that one of the goals of the Ivory Paleo Ecology Lab is to learn what the vegetation once looked like on the landscape in parts of Asia. They determine what plants were there and their abundances by conducting scientific experiments. But what if Katie and Dr. Ivory were able to send scientists back in time to determine what species were there instead? Would that be easier? Think about that for a moment. I personally think it would be way easier if Katie could travel through time. So today, we will become scientists of the future and survey our own backyards by doing a mini Bioblitz. A Bioblitz is an activity scientists do to figure out how many different species live in a certain area by investigating and using references such as field guides or the internet. By studying an area, you can be a scientist like Katie and determine what plants and animals are present. Maybe someday other future scientists will thank you for your work. You're gonna need some different materials to do your very own Bioblitz. So for this activity, you're gonna need appropriate outdoor clothing and footwear, and you're going to need to bring some water with you and a way to keep track of your species. I like using a notebook and a pencil, but you can use your phone if you want. And I like to not only keep track of my species, but also the date and the time and the weather and temperature out. So feel free to write that. You can also use some great guides to help identify some of your species. I love using my bird field guide, and there's also a lot of different apps on your phone that you can use to help identify plants and animals and anything else you can find. So I'm also going to bring a phone for the camera and to help me identify things. And then I'm also going to bring my handy binoculars so that I can see birds far away and anything else I can find. Looking cool is optional, but encouraged. Now that you have your gear, it is time to start your Bioblitz. So step one is you need to assemble your team. So this can include friends, pets, siblings, parents, whoever you want to help you with your Bioblitz. Okay, Perry, I need you to find me some plants. Make sure you have an adult for supervision purposes. Step two, define your area of study. Whether it's your backyard, a local park, or a tiny patch of grass, make sure you know what area you'll be doing your Bioblitz in. And if you have a tiny backyard like me, feel free to add some of your own creatures to find. This is Lumpy. He's a frog. Step three, go outside and identify living creatures. What plants can you find? Do you see any animal footprints? What kind of trees are there? Do you see any birds flying around? You can ask yourself these questions and more as you explore your area and collect data for future scientists. To demonstrate what a Bioblitz might look like, let's check out scientist Tim and see how he's doing.
Hi campers, this is Tim from Science U at Penn State. And for today's science camp, we are going to be doing a bioassay or walking around and seeing how many different species we can identify. So for today, we are at Hartley Wood at the Arboretum at Penn State, and we are going to talk about some of the species that you can find and also different methods to identify them. Now, a couple of things to keep in mind if you're going to be doing this activity. If you are out in the woods, make sure that you have a parent or guardian or an adult with you. There's all sorts of ways to get lost. Make sure you only go places you know. This is somewhere I'm pretty familiar with. Also, make sure you watch out as you're walking around for brush like this. Ticks love brush like that, so make sure whenever you get out of the woods, we'll do a tick check on our legs and everything to make sure that no one's getting Lyme disease today. We're going to start off with a plant that everyone always asks about and many people are familiar with, and you can find it along trails like this all over Pennsylvania. Now you'll be able to identify it based off of the 3 leaves. It has kind of a little bit of a waxy tint to it. It looks a little bit reddish. This one is poison ivy. Now there's a lot of 3-leaf things that people can get confused with poison ivy. One that I hear lots of people say is actually sitting right here next to it. This guy's got 5 leaves. People get confused. This one's safe to touch with 5 leaves. This is Virginia creeper. Virginia creeper is a native plant that has, you don't need to worry about this at all. It climbs trees the same way that poison ivy does, but it does not have the same sort of effects that poison ivy does. Now, one thing about poison ivy is that you may have touched poison ivy before, and you may not have gotten a rash. I have done it by accident all the time, but eventually at some point in your life, you might suddenly become sensitive to it and become allergic. That happened to my dad whenever he was working for a landscape company. Just one day got rashes all over him. So that's certainly one to watch out for. One of the first things that everyone wants to learn how to identify is trees, because they're big, they're everywhere, and in Pennsylvania, there's not actually that many common species, less than 100. So it's pretty easy to learn a large portion of them. So we'll start off with a couple here. One that is very easy to identify based off of its very white, light-colored, flaky bark is the white oak. So white oaks, there are some massive ones out here at Hartley Wood. And white oaks tend to be tall, they tend to be somewhat broccoli-shaped in my opinion. And sometimes they have branches like this guy up here that point almost straight out from the trunks. Oaks like to do that a lot. I don't know if we'll be able to find a leaf around here because they're so high up, but once I find one, I'll show that to you as well. Here we have some nice leaves underneath here. I'm gonna avoid the poison ivy, but here's a good white oak leaf from last year. So white oak leaves tend to have these lobes on the edge. They aren't sharp on the edges like red oaks are. And you'll have anywhere maybe 5 or 7 of those lobes on the side. There's other similar trees to white oak like chestnut oak that have many more lobes. In general, you can divide the oaks up into the white oak categories and also the red oak categories. Chestnut's another one of the white oaks. So how do we get started identifying things in nature? Look how many plants there are out here. Let's listen for a second at how many birds. Hopefully you can hear those. There must be 6 or 8 different kinds of birds right now. One way that I like to start off identifying things where I have no clue is with technology. There's actually a lot of neat apps that you can get these days. For example, I'm going to use iNaturalist here, and we're going to try to figure out what this plant is. Is anyone familiar with this plant? It's got 1 or 2 big leaves on top. That one back there has a berry on the bottom. They would have had big white flowers earlier this spring, but let's say I didn't know what this was, and I wanted to use an app. We're gonna try iNaturalist here. We're gonna take a photo. We'll use the photo, and let's see what it says. And there we go, just like that, it tells us what sort of plant we have. Now I'm going to sound a little bit like an old timer here for a second. I don't necessarily trust these apps for anything where I really need to know what it was. Let's say that someone I knew ate a plant and I wanted to identify it real quick. I'm not going to use these for that. That Mayapple, that app first told me it was a jack in the pulpit and then some sort of house plant before it finally got there, but most of the time they're pretty accurate, but just use it for your own curiosity. There are more than just plants in the woods, though. If you look really closely at the ground, I find some of the most interesting things are actually the very smallest. So we're gonna crawl down here and look in the bark mulch on this trail, and you'll see something really cool. Look at these little tiny things on the ground. If we look down inside one of them, you'll see it almost looks like a bird's nest with tiny little eggs in the bottom. These actually aren't bird's nests, but they look very similar. These are called bird's nest fungi, and they're everywhere. I would encourage you, whenever you go outside, don't just look at the really large, exciting, the trees, the birds, the deer, that sort of thing. Also take a look at some of the smallest things that you can find, because they can be some of the most interesting. Continuing with our fungi, let's look down here at this log. I found something really cool. I think you're gonna like this one. This is what we call a coral mushroom. In particular, this one is a crown-tipped coral. You can tell because the very tips have little tiny tips on the tips. Now, these grow on logs, and you'll be able to find them anywhere that you look on hardwood logs around here. Recommend checking those out. Another tiny thing for you. We'll take a look at a couple more mushrooms here. We've got a couple over here that I'm actually not even sure what these are. I think this one's close to the genus Amanita, but I'm not certain. But I do know this one over here. This one, can you see it yet? This one is what's called a wine cap mushroom. And these are pretty common out here at the Arboretum. We're having a hard time finding it. There we go. Now, wine cap mushrooms can actually get much larger than this. I'm not going to flip this one over because I don't want to destroy it, but I saw some back here that I can show you. They are normally like a wine blush red on top, or a little bit lighter. They have that weird stem, and if we go back here to see some that already got kicked over, remember, never destroy anything that you find. They have these weird purple gills. Look underneath there. Get that to focus for you. There we go. They're a dark purple color. Now that's really unique among mushrooms, and remember, never eat any mushrooms that you find that haven't been identified by an expert, but those are one that a lot of people do like to eat. Before we head back to campus, I would like to talk to you about identifying animals. Now this is hard. Looking around, I hear things running through the underbrush. I hear birds, but when you see them, it's only for a second. The mushrooms and the plants, they didn't run away. The animals, they like to run away. So one thing that we can do to identify animals is listen to their calls. Let's listen in for a second. Now there are lots of websites. Cornell has a great one that will let you identify bird songs, and they have apps. I would recommend those. While you can find birds with binoculars, they fly away really quickly, and sometimes the song is what will trip you off to let you know that, hey, there's some cool bird around here. So there's our activity for this week. I'd recommend that you guys try it. Go out in the woods or a park, whatever's near you. If you're downtown, maybe you have a park, and try to identify as many species as you can. Remember a few tips. You can use dichotomous keys, and we'll throw some in the resources section, either on our website or on WPSU's. Or you can download some apps. iNaturalist is a great one. Snapchat even has a scan feature. There's all sorts of wonderful ways you can get out there and start identifying things in nature.
Thanks, Scientist Tim. Are you looking to take your Bioblitz a step further? Check out these cool extension activities you can do to make the most out of your time in your area of study. Which of these do you think is a more effective way to learn about a new species during your Bioblitz? A flower. Cool. Bye. I don't know about you, but I think actually studying the flower would probably be a better way to go. And I know what you're thinking. Studying. Gross. Now, this isn't school. It's not like studying like that. It's just getting to know the flower a little bit better. And I'm going to teach you a way to study the plant, but in a really fun way. And that's called nature journaling. So I can really look at this flower, and I'm going to draw a picture of it in the notebook I brought. That way I'll be more likely to remember the different parts. Another activity you can do is take pictures throughout the year and see how your area of study changes over time. Seasons, weather, and time of day can all change what types of plants, animals, and fungi you can find. Try taking a picture every day this week and see what changes. And after that, try taking a picture every season and see how many differences you can spot. Thanks for learning with Sarah and I about her Paleoecology lab and the cool work she does to study the different levels of vegetation over time. And thank you for doing a Bioblitz with me. I hope you had fun. Thanks!